Welcome to the Proteomics in Proximity podcast, where your co-hosts Dale Yuzuki, Cindy Lawley and Sarantis Clamidus from Olink Proteomics talk about the intersection of proteomics with genomics for drug target discovery, the application of proteomics to reveal disease biomarkers, and current trends in using proteomics to unlock biological mechanisms. Here we have your hosts, Dale, Cindy and Sarantis.
Welcome to Proteomics in Proximity. I am Dale Yuzuki, your host, with my two co-hosts, Sarantis and Cindy. Say hi.
Hey there.
Hello there. Nice to see you.
For our inaugural episode, yes, the very first episode, we'd like to go ahead and have you, the audience, know a little bit more about us. And Sarantis, would you mind going first and telling us a little bit about your background?
Yes, thank you. Thank you, Dale.
Yes, tell us.
I just start my bachelor and my PhD in Italy, in South Italy, studying Drosophila genetics and epigenetics.
Oh, and that's
then,
great.
He's a real geneticist. He's a real geneticist.
And then I moved to Max Planck Institute for Epigenetics in Germany, where I joined the lab of Asif Akhtar, the director of the institute, working on chromatin, gene regulation, and nuclear functions. And this
Wait a minute.
Yes.
Max Planck has a center for epigenetics?
Yes, it was immunobiology and epigenetics, and it was, I think, one of the first worldwide nominated epigenetics institutes, actually. And I was really happy, and I was really lucky because apart from Asifa, it was Thomas Genovine also there, the famous epigeneticist, Super 39, the code, let's say, epigenetics code, and it was really, really nice times when the epigenetics was emerging in the gene regulation field. And after staying for quite a long time, scientist.
How long's a long time?
A little bit more than 7 years I stayed in Max Planck, actually. And then I joined industry, Active Motif, doing a consultant for epigenetics project. And since December, I am part of the great Olink team as a scientific director for multiomics, where I try to match the proteomics and preach the importance of proteomics in the multiomics world. And I'm really glad and happy to see how people, they like proteomics and how multiomics world is expanding. Yeah.
And affairs of the heart.
Yeah.
Right, Sarantis.
It's true also.
The affairs of the heart referring to, Cindy?
Cardiology, cardiometabolic, that sort of broad umbrella of disease types that I think Sarantis has learned a lot about.
I see.
But so Sarantis, what led you to industry? What made you decide to move from academics?
That's a great question. I'm always asking myself also, why this happened? Yeah, and it's I think the motivation and the internal question to learn more and to get to know about novel technologies. I think at the point in the academy I have reached my top level. And I wanted to explore more fields. I want to explore technologies and how this can be applied to the day-by-day basis, to the disease areas, right? And I think the Olink offers me this possibility to apply my basic knowledge in science to application, to translational medicine, and to biomarker discovery. And I can tell you it's a great journey already.
Well, we're certainly lucky to have you. That's for sure. Very lucky to have you.
Was that a hard transition?
Lucky to have you too.
Was that a hard transition?
No, actually, not Doyle at all. Because in industries we are doing science, and we're doing the high-profile science, and we are dealing with high technologies and novel technologies. And it was really smooth. I feel I never left academy because I'm always reading papers. I'm trying to be updated for the novel technologies and discussing with scientists. I'm discussing about projects, and I'm bringing the value to the scientific life, and that's really amazing. It's really exciting. I haven't seen any change in my daily life. That's for sure.
I see. So, and officially, your title at Olink is
a scientific director for multiomics, and I'm also taking care of cardiometabolic disease research areas. Yeah.
Yes, I see. But this is scientific affairs, which is a unique discipline within commercial activities. Yeah.
Yeah. I mean, it's I'm the bridge commercial, R&D, marketing. My goal is to preach about the use of proteomics to the multiomics world and to match the basic research with the translational medicine research. That's my goal, actually.
Yeah. And getting back to your work at Max Planck, as well as Active Motif, was the main method of epigenetics looking at 5-methylcytosine, or were you looking at also histone modifications and the whole?
I mean, we are focusing mainly on histone modifications, transcription factor bindings. Of course, we have done studies with methylation, but our main focus is histone modification, histone code, accessibility of the chromatin, and how this may interfere with the gene expression, and how this regulate the gene expression. This was the main focus.
Yeah.
And it's a really nice connection to the mechanistic side, of the nucleus, because you can see accessibility of chromatin and connect it to the gene expression. And now coming from the proteins, side, you get the real phenotype, right? And that's
you get what
match.
all of those upstream aspects have affected, right? Which is exactly what I'm attracted to around proteomics as well. That's great.
That's great.
In all that work around gene expression, was there concern that the relationship between RNA presence or absence, with all that complex epigenetics upstream of that connection between the presence of RNA and the actual protein, was that ever a concern of researchers in terms of that linkage?
No, I haven't seen the concern, and I don't think that's a concern, because it's question-based, scientific-based, depends on the scientific question that you have, right? But there's a complementarity. There's a complementarity because you learn different things from proteomics, you learn different things for transcriptomics. They are quite an overlap, not really nice correlation, not really high correlation to be correct, but this is normal because this is biology. There is a lot of steps from regulation to translation, to protein translation. There's steps of point transcription regulation for epigenetics or epitranscriptomics, but this is also the beauty of science. In order to get a better vision and connect better genetics to phenotypes, I think you have to apply both transcriptomics, proteomics, and actually you have to apply multi-omics approaches, right? That's the really beauty of science.
Well, and we've seen this transition, of course, from bulk RNA as the technology has evolved from bulk RNA sequencing to single cell sequencing, which I think is enormously helpful in understanding mechanistic biology. So I think, RNA is getting at real-time biology, and I think proteomics is as well. Yeah, that's so interesting, that background. Thanks,
Single
Sarantis.
cell is where the entire field is going. And there's so much interesting biology to reveal. Thank you, Sarantis. I know your background in epigenetics, it gives a unique perspective on this particular podcast. Appreciate it.
It really does.
And Cindy, what about yourself?
So background? Sure. So I did my undergrad in biopsychology. So I thought I'd be in neurology. I thought I'd work in that field. I think I somewhere in my final year, maybe my junior year, I realized that the tools were hard to implement. It was just really hard to get at what's happening in the brain, and so I just started looking around for what was it about biology that was so fascinating to me. And so I actually have a similar background to you. I taught high school for a little bit while I figured that out, and then I went back for evolutionary biology. So I became, gained clarity, I should say, around my fascination with understanding how we reconstruct what's happened in the past to understand systems today. And ultimately did my PhD in a biological system in the ocean. So worked for fisheries. Ended up working for fisheries for 10 years. Look, using genetics as tools. So I make the comment about Sarantis being a pure geneticist, right? Because calling me a geneticist when I'm really just using genetics as a tool, I've thought about that a lot. But I guess we're all pushing the field forward, right?
So wait, when you talk about fisheries, you're talking about places like in Maine or down in Florida? Help
or
us.
Gloucester, right? Or the coast of California, or Newfoundland, or certainly Iceland,
Wow.
Norway. Yeah.
Wow.
So your work took you, or your research took you to far-flung places.
I was really lucky. I was really fortunate.
Well, were you then involved in some of the grittier, dirtier aspects of the fisheries industry? By that I mean, I can just imagine.
Did I go on boats?
Yeah, yeah. Specifically, that's what I'm interested in.
I think I remember after my presentation for my dissertation, somebody said to me, "So you just did one cruise?" And I was like, "Oh, I made a mistake. I really should have emphasized how many cruises. I think there were 8 cruises that contributed in some way to the final dissertation."
They would call it a cruise, literally.
We call it a cruise. Yeah.
My association with cruises.
Yeah. In fact, we had cruise directors, right? So you think about the Love Boat or some, show when you're a kid and think,
but then
very
you're
different.
collecting samples. Are you collecting samples then in your second?
That's right.
On boat, or but you have it on? Oh, that's
so
great.
Both adult samples in some cruises, that was the focus, and so there was actually a lot of scuba. So working off of those NOAA ships. But also larval samples. So it turns out with some of these fish that are bottom dwellers, that you can't just collect them as larvae along the bottom. They're actually pelagic. They actually move through the water column as they develop and then eventually settle into places like kelp beds, depending upon the species. And so we needed to understand their life history. It turns out that, people might not think about this. I didn't before I worked for the fisheries, but the burden of demonstrating that we're overfishing is on the managers of the fishery. And so collecting the information to demonstrate that is essential, especially if you want buy-in from the fishermen whose livelihood depends upon being able to have access to those fishery sites. So a big part of my dissertation was looking at marine protected areas and characterizing the scale at which they could help reseed the fishery outside the protected area.
Yeah, that's cool.
Wow. You've been places. You've seen things.
So have you. You both have, right? I mean, I think I just have to emphasize that Cervantes wins the prize for knowing the most languages in this group.
How so?
Yeah.
Cervantes, how many languages do you know?
Only three for the moment, and German a little bit, and I apologize again to my German friends because I never managed to pick up German.
I think you're conversational in German at the very least, but yeah.
Yeah, well, I got to give Cervantes the most languages prize. Good
there
for
we go.
you.
There we go. And I get the most cruises prize, maybe.
There you go. Definitely.
That's for sure.
Then how did you end up in industry, Cindy?
I transitioned to industry. It was a big surprise to me. I had been suggested for a position at this little company that I thought was probably, it had a lawsuit against it. I had a friend who worked there, this little tiny startup. I thought it was tiny, about 150 people. I guess that's mid-sized these days. But and so she said, this job would be perfect for you. And I said, I'm really looking for something in academic. And I had my eye on a couple of postdoc positions along the coast of the U.S. on the West Coast here. And I said, well, I'll go interview just for the practice and I'll learn a little bit. And I was so blown away by the technology and just the ability to support technology that is a rising tide that lifts all boats. Like that just blew my mind. And it felt like an opportunity to learn so much about so many different fields. And I think after having been, you know what it's like in a PhD, after having had your nose to the grindstone in a system and learning it to the extent you need to be credible in getting that PhD, this was just so different and so it's just so awe-inspiring. I was really excited about the technology.
And Cindy, what was the company?
Well, you were there.
That's why I asked. You didn't tell me what the name of the company is.
It was Illumina, and I ended up staying for 14 years. So it was such a good time.
So you joined in the very early days. This is 2004? Right. Is that right?
2004. That's right. Yeah, I remember trying to negotiate. You're coming out of a PhD into industry, like you got nowhere to negotiate from, right? So I was trying to negotiate a little extra time before I started in the position, and I got 2 weeks from the time I defended my dissertation to the time that I remember my mother and I went to travel in New Mexico. It was a fantastic trip, but yeah, I started, almost right away. It was quite a.
New Mexico was how you spent those 2 weeks.
Right. Yes. Sorry. Yeah.
Okay. And
but I was born in New Mexico, so.
Oh, okay.
I'm guessing that there were a lot of DNA sequencing at the time. What was the most weird piece that you were sequencing at the time? What was the most weird approach that you had? One of your first, let's say.
One of the first projects?
Yeah.
So I'll tell you, one of my first projects was working with Decode Genetics. Wow. And so I remember, and I had no idea the impact they had on, steering or anticipating, I should say, where the field might go. They're very...
Sarantis, to clarify, right? This is before NGS. So at the time, Illumina was just offering genotyping.
That's right. So it was...
Yeah. So 2003, Illumina launched an 1152-plex Golden Gate genotyping platform. And then Illumina was able to sew up a lot of the HapMap projects that were going on. And this was, again, after the Genome Project, right, to characterize variation across populations. And thus, Cindy, your first role was it a project manager? Or?
Yeah, I was a project manager within the scientific team that delivered data to customers that were sort of testing out the technology in order to determine whether they wanted to invest in a bead lab or a bead array reader. Now, the bead lab was the big genome center, offering by
$1 million.
Illumina.
It was $1 million sort of LIMS, liquid handling automation system.
Yeah. It was... I had not worked at that scale before, so seeing the number of samples and the number of variants within the genome that were queryable at that point was just mind-boggling to me. And the person that interviewed me was just such a great people person. He just listened to all my, objections or all my, questions about the technology. He was so patient, and I thought, I could work for this person.
Who was it, by the way?
John Stolpnagel.
Oh, okay. One of the founders of Illumina.
That's right. He was very, just a great leader, just a great person to learn from.
And for those of you who may not be familiar with the name, he is the principal behind many, many companies after Illumina.
Successful companies.
Many successful companies.
Yeah,
correct.
Yeah.
And what's I find fascinating, right, is at that time, 1152-plex blew people's minds as far as how many genotypes you can get at the time, because people were used to like mass spec methods, right? From what is that San Diego company whose name I can't remember? Sequinome, right?
Yeah.
It was a handful, maybe 10 or 15 SNPs at a time per sample. And you go from 15 at a time to over 1,000. And then, of course, the first human one genotyping bead chip, which I was involved in the development of that, was 108,000. So we go from 1,100 to 108,000 to the first HapMap chip that was over.
Remember, we had the gene chip in between. We had a 10,000 chip in between.
Oh, I don't remember that.
That was gene-centric, yeah.
Okay. And then there was a HapMap chip. Yeah. Uh-huh.
Yeah, but then really quickly that 300, the HapMap 300, that was tag-based SNP selection, intentional SNP selection to collect additional information beyond just the ones you're querying, because you have some understanding of genomic diversity within the population, that quickly overshadowed that gene-centric chip. But I thought that gene-centric chip was, maybe it was 100,000. Maybe that's the one you're thinking of too.
What's interesting is the parallels to today, right? where our Olink's competitors on the low plex side, they do 4-plex, they do 10-plex, they do 20-plex. And now Olink comes out with 96-plex, and then panels of 96, and then 1536, and then now 3000. Okay, may not have ramped as quickly due to the inherent challenges, right, of very specific
complexity of proteins. They have a dynamic range, those little buggers. Yeah.
Yeah. And it's fascinating to think, well, we're on a similar multiplexing track here. And, right, it's history is prologue. It really does give us ideas of where the future is.
Yeah, and I think we can think about it quite similar to genotyping, right? Targeted locations in the genome. It's a great way to get a general view of the whole genome. It's sort of like a satellite view of the genome before sequencing technologies evolved to be so accessible and so affordable. And I think we're going to hopefully we see these technologies on the horizon that may offer a future of next generation proteomics, or maybe next generation proteomics, that maybe one day we'll be able to sequence the proteome. I think that mass spec is beautiful because you can see everything, but you're limited by how much you can push through. So those low abundant proteins are really, really challenging with that technology. So I think that's where we're nicely complementary to existing methods.
I mean, here it is.
This, Cindy, how was the transition for how do you see genomics to proteomics? How was this transition actually for you?
Yeah, good question. So, I was pretty clear about what was motivating to me about the genotyping technology, and then ultimately the sequencing technology. I stuck around, holding that tiger by the tail while it, it blew into the Illumina that it is today. And I really wanted a way to understand the impact genetics is having on more real-time health. And so I actually had a stint at a company called Metabolon, looking at metabolomics. Now talk about complexity of biological pathways, right? And I was there until I saw the launch of the NGS readout on the proteomics at Olink. And that NGS readout was an aha for me, and I thought, wow, I can help with that, and I'm excited about the specificity, the quality of the assay, which I think is exciting, because you want to be able to make discoveries and then drill into those discoveries and focus in on individual targets, right? So that was the attraction. And I actually approached Olink and said, you should hire me.
Just like that.
So cheeky, right? Never in the million years would I expect myself to have done that. But they were very amenable. I had a great experience with that. And I will say, working for a Swedish company with a U.S. representation is a really nice. It blends some nice qualities.
It's remarkable. Before I joined Illumina in 2003, I was at QIAGEN, a German company, for 4 years, representing their commercial efforts. And this is, of course, 1999 to 2003, where I saw through the whole genome project from the lens of a sample prep provider for the Human Genome Project. Of course, at that time, Affymetrix had just grown like gangbusters, and all this interest in whole genome transcription, expression analysis via microarrays. And coming back to a company that's from Northern Europe, and just the precision in QIAGEN is wonderful because the engineering mentality is very much exhibited, right? The precision of their of the assay development and the product development. And at Olink, I see shades of that as far as very disciplined approach to accuracy, to the quality of the product. There's a lot of care taken, right?
And it's great cross-functional collaboration here at the company. Yeah. And I think we all know that more contribution you get from diverse opinions, the better your product's going to be, provided you also have that discipline to ensure that you've got the specificity. So anyway, It's a fun ride. Sarantis, I'm curious what you're what you're most excited about in the coming year at Olink in your role.
I am really excited to see the data integration of really multi-omics data coming through, because we hear a lot of multi-omics, we hear a lot of this buzzword multi-omics, but we are not still there due to different reasons. And I think Olink offers the perfect tool, being an NGS-based assay mainly, to create this multi-omics approach in life, to bring it in life. And I'm really excited to see projects coming, really multi-omics projects that they have epigenetics, they have transcriptomics, they have proteomics, they have genomics. That will be really the future of science.
What do you think has held that back so far? Like, why do you think now is the time, right? I have this sense that we're in an inflection point, right? There's this energy when I go to conferences, for sure. And so I'm curious, your perspective on that, Sarantis.
No, it's just I'm really happy to have your feedback on that, because it's really an open discussion. I would really like to know from your side, from your perspective, how do you see this? I think that people, in regards to proteomics, the main, assay was mass spec, that it was really difficult to bring it in the multi-omics world. I think having an NGS-based approach, this makes things easier in this respect. Big data. Having expensive experiments, the sequencing is not an easy experiment to do. You need a bioinformatics tools. You need bioinformatic analysis. You need the specific platforms that they can integrate this data, that they are not well advanced. Let's say, and they're
data
really,
analysis.
they are not so well advanced in the sense that they are really not easy accessible to everybody. In this respect, of course, they're advanced, but not. The science has to be easy, accessible to everybody. It has to be easy for everybody at the end. And we are not there yet for the multi-omics approach, but we hope so.
Yeah,
what do you think from your side then?
Yeah, I was just going to say, I think you touched right on what I come across a lot is this analysis piece, and having tools to make those analyses happen, where you're integrating these data, because some of the platforms allow the data to sit side by side, but actually having scripts and tools that are bringing those data together in a multi-omics analysis is not trivial. But I think maybe part of as you were talking, I was thinking maybe part of this sense of urgency and excitement is all that's happened in the UK Biobank in the last 5 years, right? The exome sequencing, the whole genome sequencing, now the proteomics that's coming out of the UK Biobank, and making those data accessible means that there's a playground for people to advance their skills to integrate those tools, and other cohort projects as well. Not just the UK Biobank, but it's certainly the buzz at ASHG every year. Sorry, go ahead.
The definition of big data, right, is volume, its velocity, and its variety. So we have a big data problem, right? You have whole genomes at scale. You've got whole transcriptomes at scale. We have whole epigenomes, and now we're going to overlay proteomes. These are different types of data in large scale, in multiple dimensions.
Yeah, and I think that back to Sarantis's comment about mass spec, I think that was the barrier maybe, is that mass spec, it's hard to get a lot of samples through under this sort of service wrapper conditions where you're controlling variability and then feeding it into what you're talking about, Dale, integrated project with 50,000, 100,000, ultimately 500,000 samples. It's
and we can't talk about mass spec as a monolithic item, right? Because of all the infinite amount of varieties of GC, LC, tandem, timsTOF. I mean, you just go down the line in terms of everything from sample prep all the way through the technology, all the way through the analysis, bottom-up proteomics, top-down proteomics, and everything in between. It gets really complicated from even the analytical chemistry side. Yeah.
It's not absolutely, it's not monolithic. It's really a lot of advancements also in the single-cell proteomics based on mass spec, right? I mean, the single-cell proteomics protocols, they are designed and done by mass spec nowadays. I think there are different questions, and there are different scientific questions that you can get, different answers from mass spec and Olink approach, PEA approach. They can really nicely complement. And that's the beauty of science, right?
Isn't it?
They are really nicely complementing, can take really nice and integrated information. So that's for sure. That's for sure.
Let's ride that tide right up. All this great stuff.
Alas, we just have a few minutes left. Is anybody going to ask me about my background?
I can ask you for your background. I wanted to ask you, the QIAGEN was your first
That was my
job after
first.
that?
That's correct. Well, before that, I was a manager of a small laboratory in Santa Monica. I worked for a PI, Dave Hoon. We worked on tumor immunology back in the late 90s when nobody worked on tumor immunology except for Don Morton there at the John Wayne Cancer Institute in Santa Monica, and then Rosenberg here at the NCI. They were the only two working on tumor immunology. And now...
Look at it now. Wow. You were a pioneer.
Incredible. And then that time at QIAGEN was fascinating because I started with customer service, technical support, 1-800-DNA-PREP. And the manager of that department is Kirk Malloy, who ended up joining Illumina at the end of 2002 and invited me to interview, invited me to take a tour. And I'll never forget the tour I had because he showed me the alligator, the vacuum box in all of its 96-well glory. And this was Cervantes, their secret sauce, Illumina's, the ability to make very inexpensive oligos at scale. And so they developed this method, and Cindy's putting up her little drawing. That's
history,
me.
the piece of history,
the
piece of history of Illumina.
Different haircut, Look
at the coincidence of the fact that we're side by side. We're right next to each other in that.
Yeah, so those were kind of, like Cindy said, kind of scary days, right? Because it was a small company that had already gone public.
We had a lawsuit. Remember, there was a lawsuit over
intellectual property.
Well, there were several lawsuits, actually. There was from another company, and I learned now that, maybe that's a badge of honor sometimes, but I didn't know that then. I thought it was going to be, the death of us. I had no idea. And then there was another lawsuit. Both of them ended up being, just indications of success, I think, when we look back. But
But I think the secret sauce, right, was not just the right technology. Really, it's the people behind the technology. It's also the creative thinking of people behind the technology in terms of what are the critical things to work on? What is the critical fundamentals of the business to make it successful? And I can say career-wise, right? I've been in a lot of different genomics companies. It was 17 years from that time, leaving QIAGEN to coming back to Olink. And the 17 years in the in-between, so much I've learned in the genomics realm. And then now to bring it back home to proteomics, bring it back to very close to common disease, very close to rare disease, very close to population health, very close to wellness, very close to aging. Do you realize the advances we've made in cancer have been remarkable because of a focus on therapeutics and prevention? Can you imagine doing the same thing to longevity and aging to make similar advances? And we're on the edge of
prevention, right? There's the potential. Yeah.
Yes. It's remarkable times we're living in from the point of view of, measuring proteins at the scale that we're talking about can have true impact on cardiovascular disease, like matters of the heart versus Cervantes, as well as population health with the UK Biobank, like you mentioned, Cindy. What an amazing resource! I mean, what is it? 500,000 healthy individuals measured from 2006 onward.
Yeah,
regularly
and not to
checking in. Go ahead.
Not to forget or to close the loop. We're right back with Decode Genetics, right? They're also very engaged. So, what led you to industry, Dale?
I think what it was an opportunity. The opportunity to apply a lot of what I know as a scientist, a lot of what I value in terms of learning about science in a new way. And that is the combination of science with business. And it's fascinating, right? Some of my favorite classes as an undergraduate were in psychology. And now I get to use that every day in a marketing role because I'm thinking about the psychology of buying behavior, the psychology of, what messages resonate, the psychology of, if somebody's searching for solutions from a search engine, what terms do they use to find Olink proteomics? I mean,
Yeah.
that's part of my day-to-day, believe it or not, is
understanding
keyword
people's
research.
motivations. Yeah, back to the people, right?
Back to the people. And I think what's fun about the holding a podcast and this proteomics and proximity is that we get to talk as people about science. We will interview people as scientists. We will go ahead and talk about papers that people have written, their conclusions, their ideas. And so I think there's going to be a lot of fun to talk about sort of papers in the context of like a journal club. I think it'll also be really interesting to bring in guests, and certainly Sarantis and Cindy, you have some ideas of who you'd like to bring on in this remote kind of personal and intimate environment. And then we can just, the three of us, talk a lot about fish or larvae or what's it like to scuba dive in Nova Scotia or wherever it is you were. Yeah. Or maybe what's it like to work for one of the leading institutes in Germany there on a true model organism from a true geneticist, no doubt, right?
Yeah. And if we can give some perspective to maybe graduate students who are finishing up their PhD around ideas of what it's like to be in industry, I'd love to contribute to that as well. I'm still pretty passionate about working with students. So.
Great.
Well, again, people, right? People.
Again, people. Well, we got to get that in there. We got to get that in there. All right. Well, until next time, take care, audience. Thanks for joining.
Thanks for joining.
Right.
Thanks, Dale.
Bye.
Thanks, Sarantis.
Thank you. Thank you, Cindy.
Thank you for listening to the Proteomics in Proximity podcast, brought to you by Olink Proteomics. To contact the hosts or for further information, simply email info@olink.com.